Hello, everyone. So this is my video report for the grant award by the Kawa Foundation. I begin by thanking the foundation for funding my research during the last year. The title of my project is Information Processing by Biochemical Enzymes. And in this brief presentation, I will outline our main achievements during this year. So the main subject of my research has been to understand the performance of biochemical enzymes that replicate information. So there are several examples of this. One are polymerases that take polymers such as DNA and they have to replicate it. This task is usually performed in biological systems with very high speed and high accuracy. And what we wanted to do is to establish a computational platform to understand the limits to the performance of the systems and think about how to apply these ideas to important applied cases of these reactions, such as the polymerase chain reaction that is widely used in, for example, in the COVID-19 tests. So the project has been very successful, and here I will outline our main achievements. So the first is about understanding the limits of performance of these machines. So in optimization theory, when something has to optimize multiple quantities, so for example, in this case, speed and accuracy, one should expect a set of optimal solution. This is usually called the Pareto front in optimization theory and is depicted in this image. So these are the set of points that constitute these fronts for which it's not possible to increase speed and accuracy at the same time. And our aim was to understand how the shape of this front for concrete examples of biochemical reactions that replicate information, such as those implemented by these machines. And this is what we did. So we implemented a computational algorithm based on the theory of optimization, and we could try to simultaneously optimize the speed, minimize error, which means maximize accuracy, and minimize dissipation. And so in this three-dimensional space formed by these three observables, there will be a two-dimensional surface of optimal Pareto optimal solutions. And the front is represented in this image. We also looked at optimal fronts between pairs of observables, so between velocity and error, dissipation and error, and velocity and dissipation. These are these three panels. And the different curves are for reactions characterized by different number of steps. So in this manner, we could characterize how increasing the number of steps, the performance of these reactions improves. So the manuscript that presents these results has been is available on the archive and has been recently submitted for publication. The second example I wanted to mention is the polymerase chain reaction. As I mentioned in my introduction, this is an extremely important reaction in biochemistry. It's perhaps the most used reaction in biochemistry, and it's at the basis of, for example, many COVID-19 tests. And what we try to see whether we can use our theory to improve the performance of this reaction. So this work combined theory and experiments. The theory was performed by our group, and the main experimental part was done by the group of Shoichi Toyabe at the University of Tohoku. And so the idea is that there are two different ways in which we can discriminate right from wrong monomers. One is based on energy differences, and one is based on a difference in kinetic activation barriers. The energetic system is what's usually implemented by this reaction, but what we try to see is whether we could implement a kinetic discrimination mechanism by using so-called blocker strands. And it turns out that this is possible and indeed improves the performance of PCR. And this is, again, has been possible via this combination of theory and experiments that is presented in this right figure, where the points are experimental results and the line are prediction of our theoretical model. So the results of this research are presented also in a manuscript that has been recently submitted and is available on the bioarchive at this address. So the last thing I want to mention is that another thing that was partially funded by the Okawa Foundation, and we are thankful for that, is a very nice hybrid workshop that we held at the Okinawan Institute of Science and Technology in June. This workshop was titled Cells, Energetics and Information, New Perspectives on Non-Equilibrium Systems. And it was a way of exchanging ideas in these interdisciplinary fields that combined non-equilibrium physics, biological physics, information theory, and more. So this was a very successful workshop, and again, we thank the Okawa Foundation for supporting it. I conclude by certainly thanking the Okawa Foundation for funding this project, thanking my unit at OIS called the Biological Complexity Unit, and my other funding sources. And thank you for your attention.